Hello, and welcome to Team Vero's presentation on CRISPR gene editing. So first off, what is CRISPR? Well, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. They are a specialized region of DNA with two distinct characteristics, the presence of nucleotide repeats and spacers, in other words, viral DNA. CRISPR is also used as an approach to genome engineering that utilizes a cell's ability to repair double-strand breaks in DNA. It was discovered that bacteria transcribe these CRISPR sequences from DNA to RNA upon viral infection. The RNA guides a nuclease to the viral DNA to cut it, providing protection against the virus. This begs the question, how does it work for protection exactly? Well, in 2007, Rodolphe Barango and his team demonstrated this mechanic by using Streptococcus thermophilus as their model. They observed that after a virus attack, new spacers, foreign viral DNA fragments, were incorporated into the CRISPR region. These researchers also manipulated the spacers, taking them out or putting in new viral DNA sequences. In this way, they were able to alter bacteria's resistance to an attack by a specific virus, thereby confirming that CRISPRs play a role in regulating bacterial immunity. It's important to note that there are two vital components of CRISPR itself, Cas protein and guide RNA. The proteins are named Cas for CRISPR associated and have endonuclease activity capable of producing double-strand breaks, i.e. breaking the DNA. In 2012, researchers showed that RNAs could be constructed to guide a Cas nuclease, typically Cas9, to any DNA sequence. They discovered a simplified and practical means of employing the CRISPR-Cas9 system by modifying two RNA components into the same transcript. The result was a single guide RNA, sgRNA. Now this sgRNA is made up of these two components, a 20 nucleotide sequence of guide RNA, gRNA, which base pairs with a non-coding trans-activating CRISPR RNA, tracer RNA. These form a double-stranded hairpin structure designed to bind to Cas9. This complex can be designed so that it will be specific to only one sequence, improving the chances that the DNA will be cut at that site and nowhere else in the genome.
To target a double-strand break in a genome, the 20 nucleotide sequence of a single guide RNA is designed to base pair to a target site, which is chosen in part because of its proximity to a trinucleotide NGG protospacer adjacent motif, also known as a PAM, shown in the figure as the orange DNA. A complex consisting of the single guide RNA and Cas9 binds the target site, and the endonuclease domains of Cas9 produce a double-strand break. Cas9 has two separate endonuclease domains that cut three nucleotides upstream of the PAM, one cutting the target strand, the other the non-target strand, as depicted by the scissors. The double-strand break is then repaired by one of two mechanisms. Homologous recombination occurs when including donor DNA, which creates precise mutations. Without donor DNA, non-homologous end joining occurs to produce small insertions, base substitutions, or small deletions. In summary, application of CRISPR-Cas9 system involves two plasmids, one which expresses the Cas9 protein and the other expresses the 20 nucleotide guide sequence. Once both have been introduced to a cell or organism, they are expressed and form the complex that produces a double-strand break. If a third plasmid is introduced containing identical sequences to those flanking the site of cleavage, a specific mutation may be incurred via homologous recombination. Otherwise, non-homologous end joining will occur, resulting in gene inactivation and/or other random mutations.
Here's a simple and creative way to remember all those steps outlined by Jessica. Many of us grew up watching the Disney movie The Parent Trap. Turns out the plot of the movie is very similar to CRISPR-Cas9. The first step of CRISPR-Cas9 is to target where the break in the double strand will be. This is similar to when Haley and Annie, the twins, reunited at camp and made a plan to make their family whole again. Just like how CRISPR-Cas9 targets a specific gene, Haley and Annie target their parents in order to get them back together. Now that CRISPR-Cas9 and the twins have a target, the next step is to make the cut. In Haley and Annie's case, that's scaring off their dad's new fiancé. With the fiancé out of the picture and the DNA successfully clipped, the angle is in sight. For Haley and Annie, that means having their dad fly across the world to win their mom back and reunite the family. For CRISPR, that's having the cell's DNA repair mechanisms repair the DBS, which gives the precise mutation needed. Cas9 has been revolutionary to cell biology research. It allows researchers to explore a vast variety of gene manipulations, such as point mutations, insertion and removal of genes, and editing DNA structure, as well as deepen researchers' understanding of things such as causality between genotype and phenotype. CRISPR-Cas9 has led to the development of many techniques used by researchers such as gene editing and knockout, transcription, repression, and activation, epigenetic modifications, large-scale genetic screenings, generation of animal models, genomic imaging, and finally, lineage tracing.
Some applications of CRISPR can be seen in a study conducted using hemophilia A patients. Hemophilia prevents coagulation in the blood, which leads to a higher than normal blood loss, turning any injury into a more serious matter. Considering that injuries can range from a paper cut to internal bleeding, hemophilia A patients have a rough life and must be very careful not to injure themselves. Through research, it was found that hemophilia A is caused by a mutation to the F8 gene. More specifically, the mutation is a deletion in exons 8 through 22 in the gene. As we know, CRISPR has the power to knock out or knock in genes, meaning that perhaps the knocked out gene can be reintroduced and obtain normal functionality. Thus, it was hypothesized that using CRISPR, perhaps the gene could be knocked in and fix the mutation. However, to ensure that the procedure was a success, pluripotency must be maintained to indicate that the cells are able to behave and function normally. Researchers used adipose tissue cells obtained from a hemophilia A patient and generated a induced pluripotent stem cell line. Then they sequenced the DNA and found the F8 mutation was present in the cells. Then came the more challenging part, inserting the gene. Researchers used a modified version of the F8 gene called BDDF8, which has been modified not to include a part of exon 14 and was additionally modified by adding a BGH region, which is a transcription stop sequence that also aids in polyadenylation or the addition of the poly A tail. They also added a neomycin resistance cassette or a NEOR region to aid in identification through G418 resistance. It is important to note that after the knock-in procedure, there's a knockout procedure where the NEOR region is cleaved out via CRE expression. The final product should look like the last line in figure 8. Finally, the entire gene is flanked by right and left homology arms. After the CRISPR procedure is complete and the gene has presumably been knocked in, researchers performed a variety of tests to ensure insertion.
So, after inserting the gene into the target site of the patient cell's genome, there needs to be a way to check whether it was inserted properly and if the new gene is being transcribed. This study used PCR and Sanger sequencing to answer these two questions. Figure B shows the resulting electrophoresis show after amplifying DNA in normal patient and transgenic cells on the far right. The bands in the top panel represent amplified DNA segments, or amplicons, that were defined by primers flanking the cas9 recognition site, which is in exon 1 of the F8 gene. We see that these segments are the same number of base pairs in wild type in patient cells. Now remember that the patient's mutation is in exons 8 through 22 of the F8 gene, which doesn't affect exon 1, so we expect these results. But the longer segments of DNA in the transgenic cells indicate that an insertion took place at the correct site. However, Sanger sequencing was required to confirm that the donor DNA was in fact properly inserted. Now, to ensure that the new gene was being properly transcribed, PCR was performed, this time using primers to amplify a sequence complementary to the F8 mRNA transcript. These results are shown in the top panel of figure K. The presence of the dark bands from transgenic cells tells us that the inserted gene is being transcribed, which we don't expect to see in the patient cells, but do expect to see in the normal cells, which is what the results show. And GAPDH is just a positive control to let us know that the PCR method being used is actually detecting mRNA from all three cells. And Sanger sequencing was performed once again, this time to ensure that the transgene was properly transcribed. It's important to remember that the goal of this experiment was to insert a modified gene into patient stem cells without actually altering the pluripotent capabilities of those stem cells, that is, their ability to differentiate. These are just results from one of many tests to make sure that the cellular function of these cells were normal. Pluripotent markers are associated with stem cells, and their expression can be measured by using real-time PCR to measure the mRNA levels of each marker. The similarity in expression of these markers between patient stem cells and the CRISPR-modified stem cells indicates that these cells may be safe for autologous cell therapy.